Hi, my name is Yeonjoo. I'm a master's student in the systems medicine program at Georgetown. This is my final project for my machine learning course, and it's on using unsupervised clustering to examine multiple classes of opioid use disorder patients. Opioid use disorder is a chronic physical and psychological dependence on opiates, leading to clinically significant impairment or distress. Biologically, it's characterized by a rewiring of the brain's pleasure centers, shown here. When a cortex has received a stimulus, such as an opioid, it sends a signal to the ventral tegmental area, which then releases dopamine into the nucleus accumbens, amygdala, and the prefrontal cortex. The nucleus accumbens then activates motor function, while the prefrontal cortex focuses attention to the pleasurable sensations brought by the stimulus. There are both genetic risk factors that can make someone more susceptible to addiction in the first place, as well as environmental factors such as accessibility to opiates and other stressful environmental situations that push people to resort to opiate use. The prevalence of OUD has significantly increased since the early 2000s, and there is currently an OUD epidemic in the United States. With the onset of the COVID-19 pandemic, opiate use has become even more widespread, likely due to the financial, medical, and social difficulties that have been amplified. Consequently, there is a huge unmet need to better understand the impacts of OUD on the brain. For my project, I utilized this gene expression omnibus transcriptomic data set of 80 samples consisting of 20 control nucleus accumbens samples, 20 control dorsolateral prefrontal cortex, 20 OUD nucleus accumbens, and 20 OUD dorsolateral prefrontal cortex samples. There are 60,676 genes total in this data set. These are the methods that I used. First, since protein coding RNA and long non-coding RNA have separate expression profiles, they were analyzed separately. So the data set was filtered for either protein coding RNA or long non-coding RNA based on gene names from Ensemble Biomart. For each subset of data was annotated with corresponding metadata containing clinical characteristics of each sample, such as OUD diagnosis and brain region. Then data exploration was conducted using differential gene expression analysis, and results were displayed using a volcano plot. Significant genes were filtered and samples were first clustered using a heat map analysis. Principal component analysis was conducted to explore the variance of samples in the dataset, and PCA results were displayed with scatter plots. Unsupervised clustering with affinity propagation was used to assess how samples clustered together based on significant genes. For each cluster, the grouped samples were extracted and clusters were further explored using differential gene expression analysis. A few clusters characterized by all or mostly control samples in either dorsolateral prefrontal cortex or nucleus accumbens brain regions were grouped together to act as a control for clusters of OUD samples, with the goal of minimizing confounding effects by brain region. Ensemble names were converted to gene names using the MyGene Python package, and top significant genes were inputted into Enrichr to assess pathway involvement in select clusters. All data analysis was carried out in Python on a Google Colab script file provided at the end of this presentation that can be readily run to generate the results from this project. For protein coding RNA, differential gene expression analysis revealed 183 significantly altered genes in OUD, with 147 upregulated and 36 downregulated genes. Heat map analysis of the samples revealed a cluster of OUD samples characterized by upregulation of several significant genes. Principal component analysis suggested large amounts of noise within the OUD and control samples, and samples were grouped by brain region rather than OUD diagnosis. The PCA accounted for approximately 46% of the variance in the data, and noise variance was 6.54. Affinity propagation, unsupervised clustering of the significant data resulted in 11 clusters. Cluster 3 only contained one sample, and cluster 6, 7, and 10 contained approximately equal amounts of OUD and control samples. The remaining clusters were further analyzed using differential expression and pathway enrichment analysis tool Enrichr. Clusters 1 and 2 each contained OUD samples only in the nucleus accumbens region, and cluster 4 contained control samples only in the nucleus accumbens region. Consequently, cluster 4 was used as a control to compare clusters 1 and 2. Differential expression analysis using this contrast revealed 229 significant genes in cluster 1 and 166 significant genes in cluster 2, with 92 of these genes overlapping between the two clusters. Strikingly, clusters 1 and 2 appeared to be characterized by significantly different pathways based on Enrichr analysis. While cluster 1 involved IL-6, JAK, STAT3, and IL-2, STAT5, and TNF-alpha and NF-kappa B signaling, shown here, cluster 2 tended to involve a variety of pathways, including nicotine addiction signaling, Wnt signaling, and the RAS pathway. Clusters 8 and 9 also contain only OUD samples, but from both dorsolateral prefrontal cortex and nucleus accumbens brain regions. On the other hand, clusters 4, 5, and 11 contain mostly control samples when combined. So the combination of clusters 4, 5, and 11 was used as a control to further analyze clusters 8 and 9. Based on this contrast, cluster 8 had 191 significant genes, while cluster 9 had 57, and 36 of these genes overlapped between the two. These two clusters appeared to be involved in largely similar inflammatory pathways, including cytokine receptor interaction, IL-6, JAK, STAT3, IL-2, STAT5, and TNF-alpha, NF-kappa B signaling. To check for any differences, differential gene expression was also examined between these two clusters, which resulted in 68 significantly altered genes in cluster 9 compared to cluster 8. Analysis of these genes using Enrichr indicated pathways involved in mineral absorption and response to metal ions, as well as some of the inflammatory pathways found previously, such as TNF-alpha and NF-kappa B signaling. Similar methods were applied for the subset of data containing only long non-coding RNA. For long non-coding RNA, there were 93 significantly altered genes in OUD, with 63 upregulated and 30 downregulated in OUD. Just as in the protein coding analysis, heat map analysis of the significant long non-coding RNA revealed general separation of a cluster of OUD samples, though it appeared to be less clear than in the protein coding RNA dataset. ECA showed that reduced dimensions accounted for a very low percentage of the variance in long non-coding RNA data at only 19%, with a noise variance of 9.06, and samples again tended to be grouped by brain region rather than OUD diagnosis. Unsupervised clustering of data using affinity propagation resulted in 9 clusters. Clusters 2 and 8 contained only 1 sample, and cluster 3 contained almost equal amounts of OUD and control samples. Clusters 1, 4, and 7 contained mostly OUD samples from both brain regions, whereas clusters 5, 6, and 9 contained mostly control samples when combined. Consequently, the combination of clusters 5, 6, and 9 were used as a control to compare clusters 1, 4, and 7. Cluster 1 contained 160 significant genes, cluster 4 contained 29 significant genes, and cluster 7 contained 22. While there was no overlap between clusters 4 and 7, there were 13 overlapping long non-coding RNA genes between clusters 1 and 4, and 11 overlapping between 1 and 7. The majority of the ensemble transcript names could not be converted to gene names using the MyGene package, likely due to a relative lack of annotation of long non-coding RNAs in general. Many of these transcripts appeared to be novel or uncharacterized. As Enrichr results were inconclusive and did not reveal significant pathway involvement, long non-coding RNA genes were manually examined for each cluster using the NCBI gene database. Cluster 1 had significant genes such as SERNA2 and MIR210HG, which are involved in microRNA regulation. Part 1, which is induced by androgens in prostate adenocarcinoma, and asthma-associated NPSR1 antisense RNA1. Cluster 2 contained LRPs, which interacts with SYNCRT to regulate mRNA metabolism and transport, and IL-21R antisense RNA1, which is involved in immune responses. Significant genes in cluster 3 included a few more antisense RNAs, such as HIF1 alpha antisense RNA3 and RhoXF1 antisense RNA1. In summary, for both protein coding and long non-coding RNA datasets, data exploration using differential gene expression heat map analysis revealed upregulation of several genes in OUD, and some separation of a portion of OUD samples compared to control. However, PCA showed that the samples tended to separate by brain region rather than OUD diagnosis. Additionally, it revealed that dimensionality reduction accounts for a very low percentage of the variants found, particularly in the long non-coding RNA dataset. This may be due to the fact that long non-coding RNAs are more specific to tissue type and are usually found in lower concentrations, leading to more noise within the long non-coding RNA dataset. Affinity propagation clustering of the samples based on significant genes resulted in 11 clusters when samples were characterized by protein coding RNA. Cluster 2 was particularly interesting as it featured characteristics different from the inflammation-related pathways present in other clusters, despite the fact that all OUD samples were from subjects who had been diagnosed with OUD for at least 5 years. This suggests that different OUD patients may respond better to different therapies and points to the importance of stratifying OUD patients. In this project, Cluster 2 was characterized by pathways involving CAMP, Wnt, cholinergic signaling, and UV response. Though it is unknown whether these results are biologically relevant to opiate addiction or are related to additional clinical or environmental factors, such as subjects' lifestyles or comorbidities, they may provide insight since OUD is a multifactorial disease. Further research should be done to better understand the significance of these mechanisms in OUD. On the other hand, long non-coding RNA filtered samples were clustered into 9 groups. 3 clusters contain mostly OUD samples. These clusters seem to feature microRNA, immune, reproductive, and epigenetic related long non-coding RNA genes, but Enrichr results remained inconclusive due to a large portion of long non-coding RNAs with unknown function and significance. However, some long non-coding RNAs were among the significant transcripts altered in OUD in the overall dataset, and there's increasing evidence that long non-coding RNAs are important in disease pathophysiology. An important future step in OUD research may be to functionally characterize a long non-coding RNA significantly differentially altered in OUD. There were several limitations in this project. There were multiple confounding factors that this study did not account for, such as varying combinations and amounts of subjects and substances in subjects' systems at the time of death, or the presence of other clinical disorders or characteristics. This could have contributed to further noise and variance within the dataset, despite the separate analysis of protein coding and long non-coding RNA. Additionally, this project only utilized one transcriptomic dataset, as OUD is a multifactorial disease that is significantly influenced by environmental and epigenetic factors. This project provides a limited view into the biological mechanism of OUD. Looking more comprehensively at other data types such as DNA methylation and microRNA data might be useful as next steps in understanding OUD mechanisms. But overall, the results in this analysis suggest that inflammatory pathways are enriched in OUD, with a subset of OUD patients experiencing other neurological changes that may require different interventions. Chronic substance abuse is known to activate various inflammatory processes that are associated with neuropathology beyond the direct action of the substance on neurotransmitter signaling. These findings may support the use of administering anti-inflammatory therapies to alleviate the cognitive and behavioral manifestations of opiate addiction And for patients that may not respond to such therapies, as is the case in many clinical trials, additional clinical, transcriptional, genomic, and epigenetic data, as well as increased characterization of long non-coding RNAs, may be valuable in providing more precise treatment. These were my references. This 8th one is a publication that originally generated and analyzed the data set used in this project. Thank you for listening. I'd like to thank Mr. Brodsky for instructing our machine learning course and for teaching us all the concepts used in my project and supporting our class, as well as Pine Biotech and the OmicsLogic program. I'd also like to thank Dr. Vasudevan, the program director for our systems medicine program, for being such a supportive advisor. And finally, also my machine learning class. I've also included the link to my Colab Python code here, which contains all the code for my project. Thank you.